Good afternoon, Nili. Good afternoon, Pooja. Good afternoon, all students. In the today's topic, we are going to study about the molecular basis of the inheritance. In the last class, we have studied about the inheritance, right? Here, in this today's class, we are going to learn in the molecular basis, that is in the DNA and the RNA basis. Okay, fine. And you all know that from this chapter is a high weightage chapter in the all over the syllabus, and you are going to get around 5 to 6 questions from this chapter. It may be related to means the for this chapter you have to be perfect with the cell chapter. In the cell chapter there is a nucleus is there, right? You have to know the details about the nucleus in that chapter. Okay, and coming to the biomolecule, in the biomolecule you have to learn about the DNA. Okay, if you are thorough with that two topics from the 11th syllabus, that is the cell chapter, nucleus, and the 11th syllabus, that is the biomolecule, means this chapter will be easy for you. Okay, so that's why you have to be you have to have the knowledge about that two chapters. Okay, fine. So let us learn about this chapter, guys. And one more thing, if you have any doubt, means you can ask me in between also, no problem. Okay, if it is related to the same topic, means I will explain you there only. Otherwise, we will discuss that in the at the last. Okay, fine. So let us talk about the structure of the DNA molecule, guys. 1 minute. Okay. First, let us talk about the DNA structure. You all know that DNA is nothing but the deoxyribonucleotide, right? And Frederick Mixture. And one more thing, guys, if you have the doubt, means you can raise your hand. Okay, then you ask me question in the message box. Then I will see. Okay, because I'm going explaining. No, we have to complete this topic. That's it. Okay, fine. And you all know that who discovered the DNA? DNA is discovered by Frederick Mixture in the 1869. So that you know that in the first cells of the WBC. Okay, 1 minute. You can tell me whether I'm audible and my screen is visible. Then we'll go. Okay. Okay. Fine. Okay, Frederick Mixture in the 1869 found the DNA, discovered the DNA in the 1869. Okay, and you know the DNA structure is given by the Watson and Crick, right? Watson and Crick in the 1953, but the X-ray diffraction by the Rosalind Franklin, right? Okay, then coming to the structure of the DNA, DNA is the double helical structure, right? Double helical structure, and the structure, the two strands of the DNA is running in the anti-parallel fashion. One is running in the 5 prime to 3 prime, 5 prime to 3 prime, and another one is running in the 3 prime to 5 prime. So this is called as the anti-parallel nature of the DNA. Okay. Okay. And then coming to the DNA structure here, you can see that each DNA, here the DNA is consisting of the nucleotides, right? The nucleotide, here you have to differentiate between the nucleoside and the nucleotide. First, we will talk about the nucleoside. Nucleoside means nitrogen bases. Nitrogen bases plus sugar is called as nucleoside. The overall, 1 minute, I will off the camera. Okay. So when we are overall, we are talking about the nucleoside. If they are given guanosine or adenosine or uracil, like if that is ending with the sin, means that will be the nucleoside, because it is consisting of only the nitrogen base and the sugar. Okay. Then coming to the nucleotide, nucleotide consisting of nitrogen bases and phosphate and sugar. Okay. So these three together we are going to call as a nucleotide. And then nitrogen bases, when we are talking about the nitrogen bases, there will be guanine. Sorry, 1 minute. Yeah. When we are talking about the nitrogen bases, there will be a purines. Okay. Purines means this purines is having the double ring. You can see that adenine and guanine will come under the purines. They are having the double ring structure. And in the adenine, you can see NH2 is there. And in the guanine, you can see O is there. This is how you can differentiate. Okay. Then coming to the pyrimidines. Coming to the pyrimidines, there will be a three will be there. That is the thymine, cytosine, and the uracil. The thymine is going to present only in the DNA, okay, and uracil is going to be present only in the RNA, okay. And the thymine we also call as methylated uracil because a methyl group is there. The same structure uracil, but methyl group is there means that we are going to call as a thymine, okay. Kindly remember this: thymine, cytosine, and the uracil. Okay, so that is about the nitrogen bases. Okay, so this is about the nitrogen bases. Then coming to the DNA structure here, you can see that DNA structure is made up of the nucleotides, not nucleoside. Okay, nucleotides. Here you can see that the nucleotides are attached here in a one turn of the DNA, one turn of the DNA that is consisting of 10 base pairs. Okay, 10 base pairs of the 10 base pairs will be there in one turn. Here you can see, and here you can see this is called as the minor groove. So here this is called as the minor groove, and the larger one we are going to call as the major groove. Okay, and the 10 base pairs of the DNA is there. The distance between the two base pairs of the DNA, distance between two base pairs, we are going to call as 0.34 nanometer. Okay, if they are asking about in the angstrom, in some statement they will give the angstrom. In that, the angstrom means 1 minute. Okay, if they are asking about the angstrom means that is the 34 angstrom. Okay, 34 angstrom. Okay, remember this. The distance between the two base pair is 0.34 nanometer. Okay, then coming to the diameter of the DNA, it is the thickness is 2 nanometer is the diameter of the DNA. If it is in the angstrom they are given me, that is 20 angstrom. Okay, kindly remember this. And the pitch of the DNA, if they are asking about the pitch of the DNA is 3.4. Okay, 3.4 nanometer is the pitch of the DNA. Okay, kindly remember this. If this is about the structure of the DNA, and you know that here the DNA, in the DNA, A is base pairs with the T by the double bond, and C is base pair with the G with the triple bond. Okay, and the Erwin Chargas given the rules also, that is the in the DNA the proportion of the A and T is equal to the proportion of the G and C. Kindly remember this statement. Okay, and this also A plus G is equal to T plus C, or A plus G divided by T plus C is equal to 1. So this is the given by the Chargaff, Erwin-Chargaff rule. Okay, the Erwin-Chargaff rule is different for the different species. Okay, and this Chargaff rule is not applicable. It is not applicable for RNA. Chargaff rule is not applicable for the RNA and also for the single stranded DNA. Remember this, guys. For the single stranded DNA and for the RNA, the Chargaff rule is not applicable. Okay, and the ratio of the adenine, guanine, and the thymine and cytosine is different in different species. Okay, it will be different in different species. Okay, remember this. We are going to get the one question from this topic. Okay, fine. Then coming to the arrangement of the DNA, how the DNA is going to be arranged in the nucleus means you all know that chromatin, right? Only during the mitosis it is going to be arranged as a chromosome. Till that it is going to be arranged as a chromatin, right? Now also it is in the chromatin. Only during the replication process, that is in the S phase, it is going to be turned into the chromosome. Fine. So here you can see that DNA, guys, the DNA is arranged into the chromatin structure. Here you can see there is a, for the formation of the chromatin or the nucleosome, we require the histone protein. So there will be a four histone proteins are there. That is the H2A, H2B, H3, and H4. So this is in the 2. Okay, means 8 histone proteins. Okay, so there will be 8 histone proteins that are arranged. Octamers we will call as the octamer. Okay, histone octamer will be there, and H1 is going to be act as a linker. Kindly remember this: H2A, H2B, H3, and H4, and H1 is going to be act as a linker. Okay, And here you can see that will be in the 2. And how much DNA it is going to be there in the 1 histone octamer means that is 200 base pair. Remember this also, 200 base pair of the DNA will be there. Okay. It is arranged into the chromatin. And now coming to the chromatin, there will be a 2 types of the chromatin. Sorry, before going to that, histone protein. This histone protein is rich in the basic amino acid. That basic amino acid is nothing but lysine and arginine. Lysine and arginine is there in the histone protein. Kindly remember that. Okay. Kindly remember that histone and the lysine is the basic amino acid which is present in the histone. Then coming to the chromatin, one is a euchromatin. Euchromatin means what? The chromatin here, the histone proteins are arranged loosely. Okay. So histone proteins are arranged. In the statement-wise questions, you will get this concept. Loosely arranged histone proteins, and because of the loosely arranged, you can see the DNA in between, right? In DNA in between. So that's why it is called as transcriptionally active. Transcriptionally active. Okay, it is transcriptionally active. Then coming to the heterochromatin, here the histones are attached very tightly. So where you cannot see the DNA, right? DNA you cannot see. So that's why it is called as transcriptionally inactive or silent. Okay. So actually in the human, there is in the females, there will be a 2 X chromosomes are there, right? Only 1 X chromosome is going to be function, whereas another X chromosome is going to become inactive. How it is going to be inactive means the heterochromatin. So it is going to be form a condensed structure that is going to be form of bar body, right? Bar body in the females. So that is going to be form a heterochromatin structure. Okay. Just remember this. Okay. Heterochromatin, where it is transcriptionally inactive. Okay. So I think, do you understood about this slide? Do you have any questions in this? Can you tell me which sugar is present in RNA and which sugar is present in DNA? Which sugar is present in DNA, guys? Which sugar is present in RNA? How can you differentiate RNA and DNA? In the RNA, the sugar is ribose. In the 2 position, in the second carbon, there is a OH is absent. OH is absent, only H is there. So that's why it is ribose. Okay. Very good, very good. Deoxyribose, very good. Fine. In the DNA, deoxyribose, whereas the RNA, it is ribose. Fine, guys, fine. Whereas in the RNA, uracil is there, whereas in the DNA, thymine is there. So that's why in the mRNA, you can see the uracil, not thymine. Okay. Fine. I think you understood this slide, miss. Kindly give me a thumbs up. Okay. So now let us understood, you'll get the question from this also. That is length of the DNA, or what is the number of the base pairs or number of the nucleotides. In the statement wise question, you will get here, get this slide. Kindly remember, in the bacteriophage, remember, it is a single stranded. It is a single stranded means, how can you get the base pairs, right? For the base pair, we require the 2 strands, right? So here it is having the single strand. So that's why it is having the 5,386 nucleotide. Okay. And then coming to the bacteriophage lambda. In the lambda, it is having the 4,000. It is a double stranded. It is having the 48,502 base pairs. Okay. Then coming to the E. coli. E. coli is having the 4.6 into 10 to the power of 6 base pairs. 4.6 into 10 to the power of 6 base pairs. Remember. Then coming to the haploid content of the human DNA is 3.3 into 10 to the power of 9. It is 9. Whereas in the E. coli, it is 6. Okay. If they ask about the diploid human DNA content, so that is 6.6 into 10 to the power of 9 base pair. Don't get confused, guys. When you are hurry for filling the MCQ in the exams, what you will do, just you will see the, okay, 3.3 into 10 to the power of 9. You have to observe whether they are given for the haploid or for the diploid. Okay, fine. So this is for the haploid 3.3. This is for the diploid 6.6 into 10 to the power of 9. Okay. In the so many exams, they will give you the find the length of the DNA. How can you find the length of the DNA means, here is the thing. the number of the base pairs into distance between the base pair. For example, we will find for the human DNA only. What is the number of the base pair in the human DNA? In the diploid. If they ask about the haploid, that also important. If they ask about the diploid, we are counting for the diploid. That is 6.6 base pair into distance between the base pair is 0.3. Right? By multiplying that, we will get that 2.2 meters. 2.2 meters. Okay. Do you have any questions here? Remember this slide. Okay. Fine. Then coming to the experiments, guys, you all know that the Griffith experiment, Griffith is given the transforming experiment. Okay. In the year 1928, that is Frederick Griffith. He's taken the Streptococcus pneumoniae. Okay. That is a bacteria, Streptococcus pneumoniae. In the Streptococcus pneumoniae, there will be two strains will be there. Okay. One is the S strain. That S strain is having the polysaccharide coating on that. For example, this is a bacteria. This bacteria is having the polysaccharide coating. Okay. So that's why it is called as a virulent. It will cause the death. Okay. It will cause the death. Then coming to the R strain, that is a non-virulent. It is lack the polysaccharide coat. It lack the polysaccharide coat. And this is non-virulent. It is a non-virulent. Okay. What he is doing is, he's taken the mice and is injecting the S strain of the Streptococcus pneumoniae bacteria. The mice is going to be die because that is virulent, because that is having the polysaccharide coat. Okay. Then he injected the SR strain to the mice. Then the mice is lived because that is not having the polysaccharide coat. That is the rough strain. That is the R strain. Okay. Then what he observed is, now he's taken the S strain and he's heat killed. When he heat killed, the polysaccharide coating is going to be removed, right? Then the S strain is injected to the mice. The mice is going to be lived in the heat killed S strain. Then again he has started to think, and he's taken the S strain and he's taken the heat killed S strain and taken the R strain. Where in the R strain there is no polysaccharide coating is there, whereas in the heat killed S strain also there is no polysaccharide coat is there. Then he injected the mice, but the mice is died. Okay, the mice is died here. So then he observed that, okay, the mice is died. Then he observed that something is going to be transformed, right? Something is going to be transferred from the S strain to the R strain. So now the S strain become this virulent strain. What is transforming here? Okay. So he stopped the experiment there only. Then again, the three scientists started the experiment. That is the Avery, MacLeod, and the McCarthy in the year 1933 to the 1944. They conducted the experiment. They continued the transforming principle. Biochemical characterization of the transforming principle is taken the heat killed S strain plus R strain. Same. Okay. Last thing. But before injecting to the mice, they've added the enzymes. Which enzymes they've added? First, they have added this RNase enzyme for the heat killed S strain plus R strain. Then this RNase enzyme. Then also mice is going to be die. Then also mice is going to be die. Okay. Then the RNA is not acting as a genetic material. Then what they have did is, then heat killed S strain, R strain, then protease. Okay. Then also mice died. Then they were told that protein is also not acting as a genetic material. Okay. Now again, they have taken the same thing. Heat killed S strain, then R strain plus DNase. Then they have seen that the mice is living. The mice is living. Yeah, the mice is living. Means what is happening here? The DNA from the heat killed S strain as damaged. Right? Then the information present for the synthesis of the polysaccharide is damaged, means it is not going to synthesize the polysaccharide coat. Right? So due to this, the mice is not going to be died. So here the DNA is going to be act as a genetic material. But it is not proved experimentally. So many scientists didn't approve this. Okay, didn't accepted that. Then again, Alfred Archie and Martha Chase conducted the experiment in the 1952 year, also very, very important. Okay. Martha Chase, what they have did is, they have taken the E. coli E. coli bacteria. Then they have infected with the radio labeled bacteriophage. Okay. So then they have seen that here you can see the bacteriophage DNA DNA is labeled with the 32 and the protein code. The protein code is there, right? That is protein code is labeled with the S35. This is important. DNA with the P32 and the protein with the S35. Then they made that bacteriophage to infect this bacteria. Then what they have observed is only the DNA is entered into the bacteria. So that's why they have proved that. But here, minute. Yeah. Then they have proved that the DNA is going to be act as a genetic material. Okay. And you know that Michelson-Stahl experiment also, they have taken this N15, they have labeled the NH4Cl and they have grown the bacteria in that media. Then they have seen that the first they have grown in the N15 media, then later they have grown in the N14 media. Okay, whenever they started the N15 media to grow the bacteria, all are labeled with the N15, then all are showed the N15. Then after the 20 minutes, in the second generation, they have grown in the second generation, they have grown in the N14 media. Okay, and then they have observed that the replicated DNA that is having the N15 and the N14. Okay. So thereby they are proved that the DNA replicated by the semi-conservative mode of replication. I think it's very easy. No need to explain again. In this method, you will get in this experiment, you will get the numerical based question, guys. Be thorough with that. Okay. Okay. So now let us go to the property of the DNA here. Why we have chosen the DNA, not the RNA as a genetic material. In some viruses, you can see the RNA as the genetic material also. But here, why DNA is very important means DNA is more stable than that of the RNA. Why means the RNA, there is a presence of OH, right? There is a presence of the OH is there. So that's why that it is in the second carbon. So that is more reactive. Then coming to the DNA and RNA, DNA is best for the storage of the genetic information, whereas the RNA is the best for transforming the genetic information. Okay, fine. Then coming to the Francis Crick, the central dogma of the molecular biology is given by the Francis Crick. Remember this also. Okay, why am I here given here means during the in the match the following type of the question, they will give the central dogma of molecular biology. So on that time, they will ask the scientist name. You have to remember the scientist name, Francis Crick. Okay. So that is nothing but the DNA. From the DNA, the information present in the DNA is used for the synthesis of the RNA. That is called as a transcription. Okay. Then coming to the information present in the RNA, it is utilized for the synthesis of the protein. That is called as the translation. Okay. And the DNA is going to be replicate, that is called as a replication. Which are the enzymes involved here for the replication of the DNA? DNA dependent DNA polymerase enzyme. Okay. And for the transcription, we require DNA dependent RNA polymerase enzyme. Okay. For the protein, for the RNA to protein, we require the enzyme that is called as the ribozyme enzyme. It is made up of RNA. Okay. So kindly remember that. And the information present in the RNA, it is utilized for the synthesis of the DNA, reverse. It is called as a reverse transcription. And for the reverse transcription, you know that reverse transcriptase enzyme is required. Okay. So kindly remember this central dogma of the molecular biology. Fine. Let us talk about the replication, guys. Okay. So now I have a question for you. Replication in the prokaryotes. Okay. And in the eukaryotes. Can you tell me, guys, in the prokaryotes, how many origin of the replication is there in the prokaryotes and how many origin of the replication there in the eukaryotes? Remember, site, origin of the replication in the prokaryotes is only one. Whereas in the site in the eukaryotes, it is having a many. Okay. So let us talk about the DNA replication, guys. Okay. So where is the DNA? In which phase of the cell cycle the DNA replication is going to be taken place? Can anyone tell me, guys, in which phase of the cell cycle the DNA replication is going to be taken place? Very good, Neeli. Very good. What about others? Yes, phase. Yes, phase. Very good, Shreya. Yes, phase. Yes, Shreya. Yes. Okay, let us understood this. Okay, kindly see the slide. Okay. In the yes phase of the replication, in the S phase of the cell cycle, the replication is going to be taken place. For example, if you take, this is the double-stranded DNA. Okay, this is a double-stranded DNA. You all know that in the bacteria, the DNA is circular. In the eukaryote, the DNA is linear, right? Where the replication is going to be start, only in the particular site, the replication is going to be start. That is going to be called as the ORI site, origin of the replication. In prokaryote, it is one. In the eukaryote, it is many. Okay. Now let us talk about the replication is started here, right? You all know that it is winded. It is winded, right? The DNA is winded like this, right? First, we have to unwind the DNA, right? For unwinding the DNA, there will be unwindase enzymes will be there. That is helicase. Okay. The helicase will unwind, okay, unwind the DNA, and now the DNA will become like this. Okay. Now the DNA will become like this. See this diagram. Okay. When the DNA is unwinded, there may be a chances of again it is winding. Okay. There may be a chances of again it is winding. For that, some protein is going to be bind to this single-stranded DNA. What is that protein is called as? Single-stranded binding protein. Okay, SSBP. That is nothing but single-stranded binding protein. Okay, so single-stranded binding protein will bind to this single-stranded DNA. And now there is another enzyme is going to come. Can anyone tell me which enzyme it is going to become, guys? Which enzyme? The primer is going to be synthesized, right? The primer is made up of RNA or DNA? Primer is made up of RNA or DNA? I need answer from you. Primer is made up of RNA or DNA? Think once again, guys. It is not the case of DNA fingerprinting, or it is not the case of polymerase chain reaction. There the primer will be DNA. In the polymerase chain reaction, primer will be DNA. So where here in the replication, primer will made up of RNA. Okay, remember. Okay, so let us go to the quick. The primer is synthesized here. The primer is made up of the RNA. Okay, so primer is synthesized by the primase enzyme. So see here, I have given here origin, then helicase will unwind, then single-stranded binding protein will bind, and primase enzyme will synthesize the primer. Okay, primer is made up of the RNA. Okay, so now our hero enzyme, that is the DNA polymerase. DNA polymerase 1, 2, and 3. Which polymerase is going to be synthesized? DNA polymerase 1, 2, and 3. Which polymerase? Which DNA polymerase? 1, 2, and 3. 3. Very good, Shreya. DNA polymerase 3 enzyme. Remember, okay? DNA polymerase 3. Okay, it is DNA polymerase 3 enzyme started. Okay. For the DNA polymerase 3 enzyme to synthesize is required the 3' OH. Okay. So that's why the primer will give the 3' OH for the DNA polymerase enzyme. Now the DNA polymerase enzyme will synthesize the DNA only in 5' to 3' direction. Okay. Only in 5' to 3' direction it will synthesize. Goes on synthesizing the DNA in 5' to 3' only. Okay. Remember. So that's why the DNA is going to be started to synthesize here. This is called as a replication 4. It is look like the Y, right? Replication 4. Here, the 3' to 5'. So see here, 3' to 5'. It is acting as a template strand. Okay, template strand. Okay, in both the thing, 3' to 5' is going to act as the template strand. And here you can see that here the DNA synthesis will be continuous in the 3' to 5' template strand. The DNA synthesis is continuous and the DNA is moving towards the replication fork. It is going toward the replication fork. Towards means it is leading strand. Leading strand is going towards the replication fork. Okay. Whereas the leading strand template is 3' to 5'. Remember. Okay. Whereas the lagging strand template is 5' to 3'. Lagging. Lagging strand template is 5' to 3', and it is lagging. It is moving away. Here you can see the lagging strand is moving away from the replication fork. Okay. Away from replication fork. Lagging strand. Lagging strand template strand is 5' to 3'. Now it is goes on synthesizing. Right. Now, once the replication is completed, all the thing is copied, right? Now what will happen? So here we know that discontinuous strand is there, right? So that's why we call that DNA replication in the eukaryotes, or the DNA replication is semi-discontinuous, because the leading strand is continuously synthesizing, whereas the lagging strand is discontinuous. No, that's why. And here Okazaki fragment will be formed here. The Okazaki fragment consisting of both RNA and the DNA. Okay. Okazaki fragment will be removed, the RNA will be removed, and the DNA will be joined by the ligase enzyme. Okay. And here also what will happen? The polymerase 1, DNA polymerase 1 will be there, right? It will remove the primer. And it will add the DNA and it will join by the ligase enzyme. The termination, kindly see the termination, what will happen? The primer will be there, right? The primer will be removed by the polymerase 1 and it will be joined by the ligase enzyme. The polymerase enzyme will remove the primer and it will synthesize the remaining DNA and it will be joined by the ligase enzyme. Okay. And about the Okazaki fragment, in the bacteria, it will be 1000 to 2000 base pair will be there, pairs will be there. Whereas coming to the eukaryotes, it will be about 100 to 200. Remember this. Okay. So this is about the replication. Replication is semi-conservative and it is a semi-discontinuous. It is synthesized by the DNA-dependent DNA polymerase. Fine. I think you understood about the DNA replication. Let us talk about the transcription, guys. Mainly the transcription, you all know that the double-stranded DNA will be there, where the one strand is going to be act as a template strand. Right? So which strand is going to be act as a template strand? Here also 3' to 5' is going to be act as a template strand. So that is going to be called as a non-coding strand. 3' to 5' is going to call as a non-coding strand, while the 5' to 3' is going to call as the coding strand. Why it is going to call as a coding strand means it will look same as the mRNA. So that's why it is going to call as a coding strand. Okay. Then coming to the enzymes in the transcription. Okay. So in the prokaryotes, there is only the one enzyme is there. One RNA polymerase is there. Whereas in the eukaryote, there will be a 3 RNA polymerases there. The RNA polymerase 1 will synthesize the rRNA. RNA polymerase 2 will synthesize the hnRNA and snRNA, whereas the RNA polymerase 3 will synthesize the tRNA and the 5S RNA. Kindly remember the RNA polymerase 2 is required for the transcription. Okay, fine. So let us talk about the transcription unit, guys. It is very, very important. You can see here the transcription unit consisting of the first is the promoter site. Promoter site is the where the initiation will start. Okay, initiation will start at the promoter site. Some of the promoter site consisting of the Tata box also. Tata box is present in the eukaryotes, whereas the Pribnow box that is present in the bacteria. Okay, and this is consisting of the promoter, structural genes, and the terminator. Okay, so the promoter region where the transcription will start and the terminator where the transcription will stop. Okay, in between there will be a genes are going to be present. Okay, and now you can see that 3' to 5' is going to be act as a template strand and the 5' to 3' is acting as the coding strand. Okay, and you know that only the one strand is going to be copied, not both the strand, because if the both the strand is going to be copied means it will form the double strand mRNA. It is going to be form the double stranded mRNA, and it is going to be from a different protein. So that's why it is not applicable. Okay, then coming to the transcriptional gene. In the bacteria, we can see the polycystonic genes. Okay, in bacteria, we can see the polycystonic gene where it is going to be a more gene. Okay, in the more gene going to be present. Only one promoter will be there, whereas the more gene is going to be there. So that's we called as the polycystonic gene that we can see in the bacteria. Monocystonic gene we can see in the eukaryote, that is also called as a split gene. Only where it is coding for the only one gene, monocystonic. Okay, fine. So let us talk about the transcription, guys. Transcription in the prokaryotes, how this is going to be started. Same, initiation, elongation, and the termination. The main enzyme here is DNA dependent RNA polymerase. The RNA polymerase 2 here we are using. Okay. So here what will happen? The RNA which is synthesized here, it is calling as a heterogeneous RNA. Okay, fine. Let us start. The RNA polymerase is going to be initiate the transcription. Means first the RNA is going to be binded by the sigma factor, because it is not going to be identify the initiation here, right? So that's why the sigma factor is going to be bind to the RNA polymerase enzyme. Sigma factor is going to be help in the identification of the Tata box. Okay, once it is identified, okay, this is called as the initiation. The initiation is going to be completed. Now the polymerase is going to be started elongation. It is goes on adding the nucleotides. Okay, goes on adding the nucleotides. Now once the termination is reached, okay, the RNA is not going to be identified. The polymerase is not going to be identify the termination region. Now the termination is going to be happen by the Rho factor. Rho factor is going to be bind to this RNA polymerase enzyme. Then it identify the termination region. Then the RNA polymerase is going to be release the RNA. Okay. See here, this is how the termination is going to be happen. Okay. For the initiation require the sigma factor. For the termination require the Rho factor. And RNA is also going to be synthesized in the 5' to 3' region only. It is going to be added the ribonucleotides. Okay. Then here there will be a complexity is there in the eukaryotes. The RNA which is produced in the prokaryotes, it is the not contain any of the exon, any of the introns or anything. Okay. But in the eukaryotes, what will happen? The primary transcript, that is the hnRNA, that contains the exons and the introns. Whereas the exon is the coding, whereas the introns are non-coding. What we have to do is we have to remove this non-coding, right? When we talk about the eukaryotes, in the eukaryotes, the RNA is going to be synthesized. Transcription is going to be happen in the nucleus, right? The protein synthesis is going to be happen in the cytoplasma, right? For that, the mRNA is going to come to the cytoplasma, right? For that, it has to be made that mRNA. So that's why there will be a 3 process will be there. One is a splicing, capping, and tailing. So what will happen? First, the mRNA is synthesized, right? That is having the hnRNA, we will call that. That is having these exons and the introns. Okay. The in between is the introns. These all are the exons. We have to remove these introns. We will remove these introns and we will join this exon. Joining of this exon, we are going to call as the splicing. Okay. Then what we will do? The capping. So for the 5' end, we are going to add the cap. That is a phosphate group we are adding. That is methyl phosphate. Methyl phosphate we are adding. Methyl phosphate we are going to add at the 5'. So that is going to be called as a capping. And for the 3', we are going to be add the adenosine. Like about how many adenosine you are going to add? That is a polyatyl. About 200 to 300. About 200 to 300 atyl we are going to add by the PAP enzyme. Polymerase. Okay, PAP enzyme we are going to add the polyatyl. So that is called as a polyadenylation. These 3 is required because the RNA now it is coming to the cytoplasma. In the cytoplasma, protein synthesis is going to be taken place. That's why. Okay, fine. So let us come to the silent features of the genetic code, guys. You all know that the genetic code is consisting of the three nucleotide. Three nucleotide. It is called as the genetic code. You have to remember the all the codons also. It is very, very important. Okay. So now let us talk about the silent features of the genetic code. You all know that it is a triplet codon and the 61 codons are there for the amino acid. There is the 20 amino acids are there. For the 20 amino acid, 61 codons are there. Where are the remaining codons? There will be a 64 codons are there, right? Remaining 3 codons is acting as the stop codons. UAA, UAG and the UGA. Okay. And AUG. AUG is acting as the start codon, and it is also coding for the methionine. Okay. And the genetic code is universal and there is no comma, full stop and no punctuation will be there. And the genetic code is degenerate, and you know that AUG is acting as a methionine and also the initiator codon. Kindly remember this genetic code. Okay. Fine. Then coming to the mRNA, you all know the different types of the mRNA is there. Messenger mRNA, which is going to act as a template for the protein synthesis, and ribosomal RNA, which is there in the ribosome, which is help for the, it is acting as the synthesis of the RNA, that is the enzyme. Okay. Then coming to the tRNA, it is going to act as a translation, which is going to be translate the message which is present in the mRNA for the protein synthesis. Okay. Fine. This tRNA is having the anticodon. tRNA is having the anticodon. Okay. Fine. Let us talk about the translation, guys. Translation is nothing but the synthesis of the protein. So in the translation, we are having the 4 steps. One is the charging of the amino acid, then initiation, then elongation and termination. So you know that here the enzyme we are using is ribozyme. Ribozyme enzyme, which is present in the larger subunit. Larger subunit, not in the smaller subunit. Okay, fine. First, what we are doing is we are going to add the amino acid for the protein synthesis, right? First, we are going to charge a tRNA. Okay. For the charging of this tRNA, we are going to add the amino acids. Okay. So we are going to add these amino acid to the tRNA. So amino acid plus tRNA, we are going to call as amino acyl tRNA. We are going to call as amino acyl tRNA. Okay. We are not going to add the amino acid directly, where we are adding the amino acid to the tRNA. Now this becomes the amino acyl tRNA. Now this amino acyl tRNA is going to be add the amino acid. Okay. Fine. Let us talk about the initiation, guys. What will happen? The first mRNA, the mRNA will come to the 1 minute. You all know that the cytoplasma. In the cytoplasma, the mRNA will come, right? The mRNA is going to have the 5' and the 3', right? For the 5' end is recognized by the ribozyme, smaller ribosome subunit, and it is binded here to the 5'. Okay. Why we require the 5' capping means for the identification. The ribosome, the smaller subunit will identify this 5' and it will come and bind to this capping. Okay. So that's why we require that. Now the smaller ribosomal subunit will come and bind here. 1 minute. Now the where is the protein synthesis going to be taken place, guys? In the cytoplasma. Cytoplasma. Okay, in the cytoplasma, the mRNA is there, 5', 3' is there. Now the smaller subunit is binded. Okay, this is the initiation codon. Okay, now for the AUG will be there, right? AUG. For the AUG, ribosome is binded. Now it is identified by the first amino acyl tRNA. Okay, the first amino acyl tRNA. Now it is come and bind to this. Okay, it is come and bind to this. What will happen? 1 minute. Okay, the first amino acid tRNA, now it is binded, right? After that, the bigger subunit is going to be bind. After the binding of the amino acid tRNA, the bigger subunit is going to be bind. Okay. In the bigger subunit, there will be a 3 site will be there. So which are those sites? Here you can see. Which are those sites? That will be a site, okay, and P site and E site. E site. So it is having the E site, P site, and the A site. Okay. So the first amino acid tRNA will bind, right? That is already binded. The first amino acid tRNA that is binded here in the P site. Okay. So now what will happen? The second amino, the first starting amino acid tRNA will come and bind to the A site. Okay. It will come and bind to the A site, and now it is going to be form a peptide bond with the P amino acid, the amino acid which is present in the P site. It will form the peptide bond. Now what will happen? This amino acid which is there in the P site, right? The amino acid tRNA that will exit, that is exit from the E site. Okay, see here, 1 minute. Okay, see here, guys. Whenever, whichever the amino acid, the first amino acid will come, right? That will bind to the A site. Okay, that will bind to the A site, and it is going to be form the bond. It is going to be form the peptide bond with the A site and the P site amino acid. Peptide bond will be formed. Now this tRNA will be remaining, right? Because the amino acid is formed the peptide bond. This tRNA deacylated tRNA will exit. It will go outside by the exit site. Okay, and again the another amino acid will come at the A site, and it will form the bond with the second amino acid, and again the second amino acid having the tRNA, right? The tRNA will be existed. The same again, the third amino acid will come along with the tRNA. The tRNA is binded along with the A amino acid, right? That will come, and again it will form the peptide bond. Now it will move to the exit side, and from the exit side it is going to be exist. Okay, so this is how the peptide bond is going to be formed. Okay, and this is going to be goes on elongating. So this is going to be goes on elongating, and this elongation is going to be stopped. It is going to be stopped at the particular site that is going to be called. For example, this is going to be formed here, where it is having the codon, that is the stop codon. Which is the stop codon, guys? UAA. UAA is the stop codon. The stop codon is not identified by the ribozyme enzyme. Now what will happen? There will be some factors will be there. Some factors will be there that is going to be bind to this stop codon. Okay, termination factors going to bind to this stop codon. Now the termination is going to be happen. Okay, release factor will be there. Okay, when the release factor is identified, it is going to be release the protein. Okay, remember this. There will be an untranslated region. In the mRNA, all the region is not going to be translated. Some will be act as a untranslated region. Okay. That untranslated region is helping for the efficiency of the translation. Okay, then coming to the 1 minute. Yeah, we are having only 10 minutes, guys. We are not going to do this gene expression. We'll solve some MCQs. Okay, kindly go through this human genome project and the regulation and also the DNA fingerprinting. That is easy. You can go through that. Let us solve some questions. Okay. In a chromosome, there is a specific DNA sequence is there. Okay, that is responsible for initiating the replication. It is called as. Can you tell me where is the initiation of the replication is going to be taken place? I'm launching the poll. 1 minute. 1 minute, 1 minute. Yeah, you can answer me in the chat box also. Yeah, which one is the correct answer? You can actually read the Human Genome Project and other things. No, guys, that's why. Very good, very good. I got the answers. So let us do one thing, guys. You can answer me in the chat box only. Instead of launching the poll, it will save the time. Okay, let us solve some more questions. Okay, so it is the ORI site. This question is asked in the 2024 exams, guys. It is called as the ORI site. No restriction site, no cloning site. Cloning site we will study in the biotechnology chapter. No recognition site also. Okay. So which of the following is not consistent with the base pairing in the double stranded DNA molecule? The number of the C equals to the number of the G. The number of the A equals to the number of the T. The amount of the purine is equal to the amount of the pyrimidine. And the ratio of the G plus C and A plus T is not same for the each, sorry, it is same for the each species. Which one is the correct answers, guys? Very good, very good. All are answered. Shashank, can you think once again? Very good, Simran. Very good, Shreya. Think once again what we studied. I already mentioned that this question we are going to solve. In the first slide only I have told, right? Yeah. The ratio of the G plus C and A plus T, it is not same in the each species. If it is same in the each species means all are having the same quantity of the DNA, right? How it is differ in the E. coli, how it is differ in the human, and how it is different in the different species, right? So the correct answer is 4. It is same in the each species. No, it is different, right? Whereas it is also correct. It is also correct where the purines are equal to the pyrimidine. Yes, because A is going to pair with the thymine, right? And whereas the G is going to be bind with forming the hydrogen bond with the cytosine with the triple bond means that is should be equal, right? Purines and equals with the pyrimidine. Okay, then coming to the next statement. In the prokaryotes, RNA polymerase is capable of catalyzing the process of elongation during the transcription. First understood, prokaryote they're asking, and RNA polymerase is asking, and catalyzing the process of transcription. Okay. Then coming to the statement 2, RNA polymerase associate transiently with the Rho factor for the initiate the transcription. Okay, what you have to understand in the statement 2, what is that factor? Rho factor. What is that step? Initiation. Is that Rho factor is going to bind to the RNA during the RNA polymerase enzyme during the initiation or during the termination? That you have to think. For the initiation, we require a sigma factor, not the Rho factor. Rho factor we require during the termination. Yes or no? Which is the correct answer? Very good, guys. Yes, yes. Yeah, very good. A is a correct, right? This is also asked in the 2024 only. Statement 1 is true, whereas the statement 2 is false. Right? Because the Rho factor is a termination factor, not the initiation factor. Okay. This is also asked in the last year only. In the eukaryote, kindly see, in eukaryote, there are 3 RNA polymerase. That is correct. In the nucleus, in addition to RNA polymerase found in the organelle, like the mitochondria is having the different and the chloroplast is having the different RNA, right? Then coming to the statement 2, all the 3 RNA polymers in the eukaryotic nucleus have the different roles. Now only we studied RNA polymerase 1, polymerase 2, polymerase 3. Now you tell me which is the correct answer. Very good, Nili. Very good. Nili only answered. What about others, guys? These all are last year questions only. Kindly see what they are telling in the eukaryotes, in the nucleus, the polymerase enzyme will be there. 3 polymerase. Polymerase 1 will synthesize the ribosomal RNA. Polymerase 2 synthesize the heterogeneous nucleus RNA. Polymerase 3 synthesize the tRNA and also 5S rRNA. Right? All RNA having the different roles. The C statement is the correct. Okay. The lactose present in the growth medium of the bacteria is transported to the cell by the action of which enzyme? Beta galactosidase, acetylase, permease, and polymerase. Very good, Simran. Very good. Neeli, now you answered for this question. What about others? Very good, Simran. Very good, Shashank. Very good, Neeli. Very good, Neeli. Yes. You all know that. Right. You all know that lac. 1 minute. Yeah. Lac operon concept. Lac operon concept. It is the polycystonic gene. Polycystronic, right? Where it is coding the Z, then Y, then A, right? Z is going to be code for beta galactosidase. Beta galactosidase. What is the function of beta galactosidase? It is going to break down the lactose into galactose and galactose plus glucose, right? What is the permease? Permease enzyme. Permease. Permease enzyme, it is going to be transport the lactose, because lactose is going to be act as an inducer, right? So that's why it has to be transported by the permease enzyme to the media, okay, into the cell by the permease enzyme. Then coming to the A, transacetylase. Transacetylase. Transacetylase is an enzyme which will activate, it is going to be activate the beta galactosidase. Beta galactosidase for the breakdown of lactose to galactose and glucose. Remember, okay, transacetylase activates the beta galactosidase enzyme. So the correct answer is permease. A transcription unit, now only we studied in the DNA, defined primarily by the three region in the DNA, right? Transcription unit consisting of mainly the three regions, right? Go back to that. Three regions, 1, 2, and 3. What are these? So where the transcription is going to be start. What is called as, and where is the transcription is going to be end. There is a middle, what is there? There is a structural gene is there. Structural gene, right? What is that, guys? We called as. Very good, Neeli. Very good, Shreya. Correct. Promoter. Yes, this is a promoter. This is a structural gene, and this is a terminator. Right? Option D is the correct answer. Okay, let us go to the next question, guys. Which of the following statement is correct regarding the process of replication in E. coli? They are asking about replication, and they are asking in E. coli. The DNA-dependent DNA polymerase catalyze the polymerization in one direction, that is 3' to 5'. Is the DNA polymerase catalyzed in 3' to 5'? Obviously no. Okay. Just to see the prime, guys. Primes you can see. And this is also given 3' to 5'. No. Okay. And here also, here they are given 5' to 3'. Just go for the two statement. The DNA dependent RNA polymerase. So here they are asking about the replication. Then here they are given the DNA. Here they are given the DNA dependent RNA. No, wrong. Here they are given DNA dependent DNA polymerase. That polymerize in 5' to 3'. Then that is correct. Because it is DNA dependent DNA polymerase, not DNA dependent RNA. Just see here, DNA and RNA. Don't get confused. By mistake, you will take. Don't do that. Okay. First, you have to see the prime. It is always synthesized in the 5' to 3' only. Okay. And it is DNA dependent DNA polymerase. When we talking about DNA dependent RNA polymerase, it is in the case of transcription. Okay. If they are given here transcription instead of replication means option B is correct. Okay. The correct answer is option D. One minute. Yeah. Okay, guys. So that's it for the today's class. If you have any question, please be there. If you have any questions, means you can ask me. Okay. So we are not able to complete the whole this chapter in this timings. Okay, whichever the important topic I have told you already. And the human genome project and fingerprinting you can study. That is very very easy. Okay. So I will go for the feedback session. You can give me a feedback. And students, if you all are in any doubt, means you can ask me in the chat box. Or if you're having any process confusion, means you can ask me. Now only I will explain that also. If you are having doubt in any replication, transcription, translation, or any process, okay, you can ask me. I will solve your doubt.